From there, things get a little more complicated. From the glomerulus and Bowman's capsule, the filtrate does go through the proximal convoluted tubule. Proximal refers to close, convoluted would be coiled, and tubule would be, well, tube. So you'd have the close coiled tube that would technically be in the cortex rather than the medulla. If you kind of trace with your finger on the glomerulus, the proximal convoluted tubule leaves the glomerulus, goes up, makes a little sharp turn down, and then goes up, and then goes back down. But that would be all part of the proximal convoluted tubule. We will talk later about tubular reabsorption, and a lot of that does happen in the proximal convoluted tubule, or at least we talked about that in lecture. I wasn't really going to mention it too much in the lab portion, but yeah, that's the part that we're talking about. From there, the filtrate does go down the loop of Henle and back up the loop of Henle. The loop of Henle is a loop to the medulla and back to the cortex. Its job is really a lot of the magic that happens in making urine. It's where the urine is made more or less concentrated. The loop of Henle, I'm not sure exactly, precisely how it works, but it's a lot like the large intestine of the digestive system. Remember, we had water being absorbed out of the large intestine, and if the remains of the food spent a long time in the large intestine, it absorbed a lot of water, and we talked about things being constipated there. If the filtrate spends a long time in the loop of Henle, that will make it more concentrated. Yeah, try not to think about it too much, but same thing on the digestive system. Also, when we talked about the large intestine, we said if food zipped through the large intestine really quickly, there wasn't time to absorb very much water. Same thing with this nephron here. If the fluid zips through the loop of Henle, it won't have enough time to absorb very much water, and that would make the urine more dilute or less concentrated, however you want to look at it. Now, I'm not really sure how your nephron knows whether to have the filtrate zip through it or go very, very slowly. I don't really know how your body determines that, but that's one of the ways you can make the urine more or less concentrated. Anatomy books and our lab book get into a little extra detail on the loop of Henle. The lab book does mention an ascending limb where it's going up and a descending limb where it's going down. There's a thick segment and a thin segment. I'm going to assume you can tell up from down and thick from thin. We're just going to call the whole darn thing a loop of Henle and go on with life. Once we do go up the loop of Henle, we'll run into a distal convoluted tubule. The distal convoluted tubule is the far, coiled tube in the cortex. Distal's far, convoluted is coiled, and tubule is still tube. So this would be the far coiled tube in the cortex. Now, do be careful. Do not get the ruler out and measure how far the distal convoluted tubule is from the glomerulus versus how far the proximal convoluted tubule is, because I'm well aware on this model, and for that matter on figure 11.4, the distal convoluted tubule is actually closer if you got the ruler out. Really, the proximal and the distal refers to the amount of plumbing that you've run through, not get the ruler out and measure it. Also, our distal convoluted tubule is not near as convoluted on our model, at least. I'm not sure about real life, but it barely just swerves back and forth, whereas that proximal convoluted tubule really took some sharp turns there. But the distal convoluted tubule is the far coiled tube in the cortex. Hint, it'll do a lot of tubular secretion for us in a few minutes. The distal convoluted tubule does lead to the collecting duct. The collecting duct, like its name says, collects urine of all things. It does end in that renal papilla, which is where we were collecting a lot of urine as well. And honestly, the renal papilla collects the urine because the collecting ducts are running right through there. while the urine does go through the collecting duct, we did have fluid coming from the glomerulus going E is for exit through the efferent arteriole. There were some other capillaries that were exchanging the oxygen and the carbon dioxide, and those do lead back to the renal vein, which takes blood back to the heart so that it can pick up oxygen and go to other places in the body. On page 114 to 115, the lab book does talk about filtration, reabsorption, and secretion. With filtration, that is the role of the glomerulus does filter the blood. Blood pressure forces fluid out of the capillaries of the glomerulus. There were some things in blood that we were concerned about. There was some water, some glucose, and some urea molecules. Filtration is a passive process, and it's a random process as well. Some of the water, glucose, and urea gets out of the glomerulus, caught by Bowman's capsule, runs through the convoluted tubules, the loop of Henle, the collecting duct, and becomes urine. Some of the water, glucose, and urea does not get filtered, goes out of that efferent arteriole, goes back to the paratubular capillaries, and eventually goes back to the renal vein and goes back to the rest of the body. That's actually a little bit of a problem. We didn't want the water and the glucose to leave the body. We want the good stuff to stay in the body. And your kidneys do have mechanisms to reabsorb the good stuff. You reclaim the water and the glucose. Usually in the proximal convoluted tubule, you can reabsorb it out of the nephron and keep it in the body. Now, any of the urea molecules that got into the glomerulus and Bowman's capsule, that's good. We wanted to keep the waste products in the nephron and get rid of that urea, so that's fine. On the other side of things, if you did get out of the efferent arteriole and you were water and glucose, that's good. We wanted to keep you. You're going to go back to the renal vein and everything's fine. But what if you were a urea molecule and you got away from the glomerulus and Bowman's capsule? I just picture this urea molecule going, "Nyeh, nyeh, 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 I escaped." Yeah, but it didn't. Your body does have mechanisms to secrete the bad stuff, like that urea that thought it was escaping, but you can actually do tubular secretion, and the bad stuff goes from the bloodstream into the nephron, and stuff like urea would be secreted into the nephron. At the bottom of page 115 is figure 11.5. At some point they do tell us, let's see, on number 4, we should study the nephron structure. We have already done the study of the nephron structure by identifying parts and pieces on the model. What we're then supposed to do is obtain a compound microscope and a slide and look at stuff under the low power and then the high power. And yeah, well, honestly, I don't usually do this one on the on-campus lab either. We just look at these pictures. There is figure 11.5, and the left side does kind of sort of maybe look like this figure here, which does have the blob in the middle, would be the glomerulus. You can see the capsular space around the glomerulus, and then what's kind of hard to tell is the one cell layer thick capsule around the outside there. But you can generally look at these things under the microscope, and they do actually look like the picture. Well, a glob in the middle being the glomerulus, and the capsule around the outside being Bowman's capsule catching the filtrate. This slide shows some of the proximal convoluted tubules and the distal convoluted tubules there. They're actually round because you're kind of looking down the tube. If you were picturing, say, a drinking straw, if you were looking straight down the straw, well, you'd see a bunch of little round things. Apparently, one of the big differences between the proximal PC tubes and the distal DC tubes is some brush border cell. Yeah, that's nice. They're all round things. They're all convoluted tubules. That's nice. On this slide, you're seeing more of the longitudinal long axis of the loop of Henle. If you were picturing our straw, instead of cutting transverse and across and about where you'd put your lips and getting a circle, if you cut it long way, yeah, I don't know why you'd cut it long ways, but picture, I don't know, cutting the banana long ways to have a banana split? That'd be the way you'd be cutting these loops of Henle, so that you would see a long loop of Henle for number 1, and a long loop of Henle for number 2, and a long loop of Henle for the third section as well. You'd be seeing most of the longitudinal up and down pieces on this slide. On pages 116 to 118 or 19 or something like that, they do start talking about the characteristics of urine or a urinalysis test. Basically, what you do is you pee in the cup, and then you take a special dipstick, you dip the stick in, and you kind of match it up with a answer key. Yeah, I know it's not an answer key, but you match it up with the answer key and try to figure out some of the values for some of the different characteristics. They do say on page 116 that to be valuable as a diagnostic tool, the urinalysis should be done within 30 minutes after the urine is voided, after you pee, or it's refrigerated. Generally, the freshly voided urine, nummy, should be generally clear and pale yellow to an amber in color. Occasionally, people will ask the embarrassing question of, what does the color have to do with anything? Generally, if your urine is a very pale yellow, you're probably drinking plenty of water. Let's face it, if you go potty and you look back in the toilet and you can't even tell you've gone, you've got some fairly dilute urine there. On the other hand, if you haven't been drinking your water and you've got rather concentrated urine and you can see where every drop landed in the toilet water, yeah, you should probably be drinking more water. But generally, the color variations do kind of indicate the relative concentration of solutes to water. They tell you about how much is in there, how much other assorted waste products are mixed in with the water. Generally, the more stuff that's in there, the deeper, the darker the color, the more concentrated things is, the more you need to be drinking your water. If you've got a lighter color urine, you're probably doing just fine. They talk about the odor and the pH and some of the other stuff. We'll go through a few of them on the next few pages. On page 118, there's chart 11.1 with some urinalysis results. Really, what you're supposed to do is dip the little stick in the tube of urine, and then you kind of compare it to the answer key, as it were. And there are several things that you can test for. It does depend on what urinalysis test strip you actually buy and get a hold of. But generally, over here on the right side is actually a normal sample. It is testing for several different things. Unfortunately, they're suffering a little bit. They're kind of hard to read. The first line that goes from kind of a blue to a green to a yellow is going to be the specific gravity. That is a bit of chemistry stuff, and it has to do with how many solutes are dissolved in your water. Again, if you've got the pale yellow color, you probably haven't got a lot dissolved in your liquid there, and you've got fairly dilute urine. That would be kind of the greenish and bluish sections on the chart. On the other hand, if you've got a yellow on the first line or an orange, that's probably pretty bad. You should probably be drinking more water. The second row with the kind of blue line there does refer to the pH. If you've got more of a orange or yellow color, that's slightly acidic. If you've got more of the green or flat out blue, that's going to be more of the basic. The third line is going to be for the leukocytes. Leukocytes are white blood cells, and you shouldn't have any white blood cells in a normal urine sample. The fourth line would be nitrites, and you should not have any nitrites in there, although you never know sometimes. The next line with the little purple arrow is going to be proteins. You should not have any proteins in there. In fact, if you have proteins, that's usually a really bad sign. So ideally, all of our dipped sticks would start out as a yellow, and they should not be green. One of the important things to test for is glucose in kind of the light blue line there. On this particular test strip, it does start as a yellow, which is good, moves to kind of a green or a really dark green. That's going to be bad. If you see a sample with a lot of sugar in it, that's probably not a good thing. Next is going to be ketones. Ideally, it should be kind of a yellowy color, although if you're dieting, you might want to see more of the purple color. Yeah, I can't pronounce that one either. Ideally, there's none of it in there. It really, nope, wrong. Urobilinogen ideally should be present. Okay, fine. It should be a normal color, though. Bilirubins would actually be some of the bile proteins from the digestive system. Those should not be in there. And then the last line is going to be blood. That really shouldn't be found in the urine either. So off to the right is a dipped stick, and it does have some nice normal values. So you can see what it should look like. We do have several samples that we have dipped a stick in for you so that you can kind of see how this would work. Sample A does have the normal greenish color for the specific gravity. It's got the decently normal pH. It doesn't have any leukocytes. It doesn't have any nitrites. The proteins, oh my God, look at that glucose. Instead of being a nice yellow and negative for glucose, oh my gosh, this one has a lot of glucose in there. You probably can't see what it says underneath it, but it does say 1,000 milligrams per milliliter, which is actually a fairly high glucose level. For our on-campus lab, we pour some sugar in a test tube, put some yellow food coloring in, and call it a urine sample. But really, it's mostly a sugar sample. All of the rest of the numbers are fine, so sample A has a high glucose, which we'll see in a few minutes is probably going to indicate diabetes. If you do eat a whole bunch of sugar, if you eat sugar cookies for breakfast and wash it down with a cherry Coke, that's going to get you some high blood glucose levels. That would also get you a matching thing for sample A. However, usually these urinalysis tests are often done at the doctor's office, and it's often a fasting test, so you haven't eaten for 8 to 12 hours before. So if you do eat the sugar cookie and drink the cherry Coke, you're probably going to have to come back again tomorrow, and they're going to want you to be hungry this time as well. But if you haven't been eating a whole bunch of sugar or drinking cherry Cokes or something like that, that might indicate diabetes. If you have been eating your sugar cookies and drinking your cherry Coke, over time your pancreas will crap out, stop producing as much insulin, and then when you do have even a regular amount of sugar, your pancreas no longer secretes the insulin, your sugar levels are still in the bloodstream, they're still high, and your kidneys will try to take over and help out. When your kidneys do that, you often excrete the glucose in the urine. It comes as a little bit of a shock sometimes to people. You peed on the stick, and now they're worried about your digestive system. Well, you peed on the stick, you had high glucose. That indicates that your pancreas is having some problems. So, yeah, they're probably worried about your digestive system in that case. Sample B here is good on the specific gravity, the pH, no leukocytes, no nitrites, protein's okay, glucose is good, ketones are fine, thoroughly unpronounceable thing is okay, bilirubin's good, blood, wait a minute, blood's not yellow, oh my gosh, we're getting some blood in the urine. That's probably due to a menstruating female. Now, if you're not a female, you're a little more worried about it. And honestly, if you are a female and not menstruating, you're probably a little more worried about it. We'll talk about some kidney damage that might be the cause of that blood in the urine. But you can kind of see how this would work. Sample C has, oh my gosh, where'd that purple come from? The purple is actually some leukocytes, which does indicate a bacterial infection. What else is going wrong on this? Oh, hey, wait, look at that first one, that specific gravity. It's got a higher than usual specific gravity. No, it's actually probably not high enough to really worry about. You probably can't read that it says 1.020, which is still in the normal range, but patient C may need to drink some more water. They're looking a little on the dehydrated side. So what does all this urinalysis stuff mean? There are actually some common problems on Table 11.1, which is on page 117. Now, unfortunately, Table 11.1 is written in some anatomy terminology, so I've kind of translated here on this PowerPoint slide. You'll want to concentrate on the things on the PowerPoint slide. They are actually written on Table 11.1 if you can read through the anatomy. Table 11.1 does have the constituents listed. That's the thing we're testing for. It's got the condition. That's generally something that's high in the urine. Look at all the terms. Almost all of them end in urea, and that's urine. So whatever's in front of it, there's too much of it in the urine. The indications are basically what's going wrong. For example, the first column or the first row does have the constituent of glucose. We're looking for high glucose. The condition would be glucosyluria, which really does mean abnormally high blood sugar or glucose levels. They have pathological and non-pathological there. Generally, non-pathological things are environmental things, not caused by a disease, whereas pathological would be some sort of disease. For example, if you do have high glucose levels in your urine, the common problem is probably high carbohydrate intake. If you were eating your sugar cookies and drinking your cherry Coke for breakfast, you're probably going to find some glucose in the urine. On the other hand, the pathological disease problem is going to be some uncontrolled diabetes. Now, we're going to concentrate on high glucose and call it indicates diabetes there. With the albumin, that would actually be a high protein. And there's quite a few things that would cause high protein. It could be some extra exertion, could be pregnant, could be eating steaks for dinner. But generally what they're really worried about is high blood pressure. Now, you might remember back from exercise 8, we called high blood pressure hypertension. So it is listed on Table 11.1 as hypertension. If you can read through that, well, that was high blood pressure. If you can't read through that, high blood pressure is certainly the non-science major term. Ketone bodies would cause a condition called ketonuria. Generally, the problem is from starvation, diets low in carbohydrates, inadequate food intake, which all force the body to use its fat storage. So concentrate on the starvation, low-carb diet. More than a few years back, there used to be some low-carb diets, some high-protein, really low-carb diets. And if you were doing it right and dieting right, what you'd do is you'd actually buy one of these urinalysis dipsticks. You'd buy several. You'd dip the stick in your urine, and you were trying to turn the ketone stuff, you were trying to turn it purple. Now, that actually worked really well for dieting because you were using fat stores, but unfortunately it also led to a few side effects like kidney disease and damage and dialysis and not good. Generally, they really don't recommend a starvation diet. If you'll cut out some calories and exercise, you'd probably do better than doing some really massive dieting. But anyway, red blood cells, the condition would be hematuria, and the non-pathological, cheap and easy explanation is a menstruating female. If you're not female or you're not menstruating, you might have to talk about irritation of urinary tract organs or infection or physical trauma. You really shouldn't get too much blood in the urine. The menstruation part would actually be coming from close anatomy, but it wouldn't really be blood in the urine. Hemoglobin would be pretty much the same sort of thing. It's just found in red blood cells. If you've got dead red blood cells, then you'd have hemoglobin in your urine. Nitrites, if you've got nitrites in the urine, that's generally a bacterial infection. The bile pigments, the bilirubinol stuff, the liver and the gallbladder do produce bile, but it goes directly into the small intestine, which goes into the large intestine, which goes out the other end. You really shouldn't have any bile pigments in the bloodstream unless you've got some serious liver problems and liver damage and you're kind of internally bleeding out in the liver. Ooh, that's not good. Yeah, bile pigments in the urine are not good. White blood cells would cause pyuria, and that's generally from a bacterial infection. If you do have some white blood cells in there, that's probably not a good thing. That's really pretty much all we've got on this chapter. Page 119 does have the same sort of stuff that was on page 118. Never mind. Page 120 has explained the significance of the following terms, and really that's this PowerPoint here where it's got some of the common problems that you might see. Concentrate on this PowerPoint. If you'd like to write it down on page 120, that works, but these are the ones we're going to ask about. Anyway, thanks for playing with exercise 11, and we'll see you next week.